Hey guys, hi, welcome back to this channel. After a long time, I guess we have had quite a long break. Am I audible and clearly visible to all of you? Please let me know. So, today we are going to start with biomolecules. The first biomolecule that we are going to read is something that is the most abundant biomolecule, that is carbohydrates. Okay, now today we'll be discussing about the important properties of carbohydrates, important types of carbohydrates, their structure and their properties. Also, a basic understanding of how to draw their structure, especially glucose, very important. Their applications, clinical and biomedical applications. So these are the basic things that we're going to cover today. A couple of MCQs I have included, summarizing both the units that we have covered, that is the respiratory chain and bioenergetics, and enzymes and proteins. Okay, so a few important MCQs. I think without further ado, let us begin with the biomolecules. Now, in the revision, before everyone else joins in, let us just quickly discuss the revision MCQs. The first one is from the first unit, that is proteins and enzymes. Now, select which of the following is incorrect. Okay, you have to select, always pay attention on what the question is asking. The question is asking for selecting the incorrect option. Okay, so select which of the following is incorrect. The first is pay attention to the options, and you have to weigh each and every option, because sometimes there can be a choice between something that is more correct and something that is lesser correct. Okay, so weigh in all the options properly. Monomeric enzymes exhibit sigmoid initial rate kinetics. Hill equation is used to perform quantitative analysis of cooperative behavior of enzymes or proteins. For most enzymes, the initial reaction velocity exhibits a hyperbolic dependence on the concentration of the substrate. The initial reaction velocity for most enzymes. So focus on the keywords. Enzymes lower the activation energy for a reaction. Now, out of all these, I think D is the most straightforward. Okay, so let me know which of these, which is the correct option, and then the correct option, that means which is the incorrect option for this, that you have to select the correct answer. So which is the correct answer out of all these options? Let me know. Yes. So the incorrect statement out of all of these is A, that is monomeric enzymes exhibit sigmoid initial rate kinetics. Okay, because sigmoid initial rate kinetics are for multimeric enzymes, like multimeric enzymes or proteins like hemoglobin, that that is a tetramer, right? And it exhibits cooperative binding. That is, once one molecule of oxygen attaches, the affinity for the other oxygen molecules drastically increases. Okay, and the other oxygen molecules can very easily come and combine to the hemoglobin molecule. So that is cooperative behavior. That is classic of tetrameric enzymes or proteins, or multimeric enzymes or proteins, and it displays sigmoid kinetics. For monomeric enzymes and proteins like myoglobin, the reaction velocity is initially directly proportional to the substrate concentration. So it is like this, right? Until it reaches all the enzyme active sites are saturated, and then it reaches this plateau. So the initial velocity exhibits a hyperbolic dependence, which is totally correct. Okay, so you should pay attention to all these keywords: hyperbolic, sigmoid, multimeric, monomeric, tetramer, monomer, myoglobin, hemoglobin. All these things are extremely important. Enzymes lower the activation energy for a reaction. Very true. This is, we have discussed in great detail. Okay, next. The flow of electrons through the respiratory chain and the production of ATP are usually tightly coupled, right? That is, both occur together. It is uncoupled by which of the following? Cyanide, hydrogen sulfide, carbon monoxide, oligomycin, or thermogenin. So I'll give you some time to mark the correct answer. So the uncoupler out of all of these is E-thermogenin. Thermogenin is present in humans in brown fat, that is on the nape and between the shoulders, especially in neonates and infants. And thermogenin, it produces heat by non-shivering thermogenesis. Okay, so what happens is the production of ATP and the flow of electrons is dissociated, so that the energy that is generated by the flow of electrons does not produce ATP, rather it dissipates as heat energy. Okay, so that is what the role of thermogenin is. Another, a poison or a drug that does it is 2,4-dinitrophenol. That is also an uncoupler. Okay, so how does cyanide inhibit the respiratory chain? Cyanide acts on complex 4, the final complex. H2S, similarly, complex 4. Carbon monoxide, complex 4. What does oligomycin do? It inhibits ATP synthase enzyme. Remember, this enzyme is embedded in the inner membrane of the mitochondria. It has an F0 and an F1 complex. Okay, so it rotates, the F0 complex rotates, and F1 is the axis around which the different globular proteins of this subunits of this enzyme are arranged. And once F0 rotates, then with each revolution, 3 molecules of ATP are synthesized. Understood? And it requires a proton gradient to function. Understood? So all these things, why? Because F0 is the proton channel. So these were the things that we discussed. Okay. So I hope with this you can go back and revise, or you must have already revised. So this is very easy for all of you. Okay, so these 2 units, they must be very clear to all of you. Now we will begin with the biomolecules. Okay. Now, for carbohydrates, what is their biomedical importance? Firstly, they are extremely polar compounds. Extremely polar compounds. Okay. Now, what is with the nomenclature carbohydrates or saccharides? So, which one is more correct, or which one is more applicable? Carbohydrates, it refers to something with the empirical formula of CH2 whole to the whole n, that means molecules of hydrates of carbon. So hydrates of carbon in varying molecules form carbohydrates, but it is not always true. Sometimes they may have other molecules as well, like nitrogen. Okay, sometimes they may be linked to proteins as well. So carbohydrate is not a universal term, or it is not an inclusive term for these biomolecules. What is more inclusive is saccharides. So we should focus on saccharides. Okay. Now, what is the difference between saccharides and sugars? Now, sugars, saccharides, what saccharides mean? They mean sugar only. But sugar has come to refer to something sweet, or basically the table sugar, that is the sucrose. Okay, so sugars might sometimes not strictly refer to all the saccharides. Therefore, the best term out of carbohydrates, saccharides, and sugars is saccharides for these biomolecules. Okay, now in nature, in plants, by the process of photosynthesis, one of the most important saccharide, that is glucose, is synthesized, and it is synthesized and stored in the form of starch, or it is used to make cellulose in the plant cell walls. These are all storage forms of glucose. In animals, mostly we take saccharides from the plants, but we can also synthesize saccharides from amino acids, and we are going to read about them in a chapter called gluconeogenesis. Genesis of glucose from a new compound or a new substrate. Okay, so this gluconeogenesis we are going to read about. So mostly in animals, we focus or we rely on the supply of glucose from the food we eat, that is from the plant material. But we can synthesize glucose also, though in limited amounts. In it is not a regular phenomena. In some circumstances, in some special circumstances, to overcome some conditions, and that is through gluconeogenesis. Understood? Next, which is the most important saccharide, that is glucose. We have already read about glucose. We are going to read a lot. Okay. Now, saccharides are the most abundant biomolecules in nature because of their reactivity. Why? Because they are extremely polar. They are extremely polar, hence they are extremely reactive, and because of their structural plasticity, that is, the same saccharide can exist in various structures. So it may have the same molecular formula, but it can exhibit a lot of isomerism. Okay, that is, its structure can vary. Its essential constitution will be the same molecular formula, but its structure might vary. About this also we are going to read later on here in this chapter itself. Okay. Next, what is the role or biomedical importance of glucose? It is the major metabolic fuel of mammals and the universal fuel of fetus, because the fetus depends solely on glucose. It is the precursor for the synthesis of all other carbohydrates in the body. That is, the storage form of glucose in animals, that is glycogen. Ribose and deoxyribose sugars that form part of RNA and DNA. Galactose, which goes on to form lactose, present in human milk. Glycolipids, present in cell membrane. Proteoglycans and glycoproteins, they form a large part of the glycosaminoglycans, and a lot of indicator molecules, a lot of carrier proteins, and they are present on the surface of RBCs as well, to provide the basis of blood grouping, and the like. Okay, so glucose is extremely important, and it is the precursor for the synthesis of all other carbohydrates in our body. That is why it is extremely important. Now, there are a few terms, especially three terms, which you should be aware of, especially from MCQ point of view. Okay, now one is glycobiology. What is glycobiology? It is the study of the role of saccharides in health and disease. So, what is the role of these carbohydrates in health and disease, in maintaining the health or in causation of disease? That is glycobiology. So, for understanding purpose, the study of or the research in diabetes mellitus can form a part of glycobiology, right? How sugar and glucose, how they are imperative in the causation of the metabolic disorder or diabetes. Okay, so glycobiology is the study of the role of saccharides in health and disease. How do saccharides play a role, or how do saccharides affect health, and how do saccharides cause disease? Next is glycome. So you must have heard of the term genome. Genome is the complement, the entire complement of genes in an organism. That is the total number of genes present in the organism. Similarly, there is a term called glycome. It is the entire complement of saccharides in an organism, be it free or be it bound saccharides. Okay, so the total amount or the total variety of saccharides that is present in the organism is what forms the glycome, similar to genome. Okay, next is glycomics. Now, glycomics, like glycome is similar to genome. Similarly, glycomics is analogous to genomics or proteomics. That is, it is the study of glycomes in all its aspects. Okay, so the study, now that you know what is the entire complement of saccharides, be it free or bound form in the organism, you have to study them, their properties, their reactions, their nature. Now, how do you study? In which field? glycomics, just like genomics. So these three terms are very important: glycobiology, glycome, and glycomics. Okay. Next, you must have heard of artificial sweeteners, right? So artificial sweeteners are nothing but they replace sucrose. They try to replace sucrose in the diet. Now, sucrose, as you know, sucrose is the table sugar I've already mentioned. Now, it contains calories. It is a disaccharide. It contains calories. And it is sweet in nature, and it contains calories. So, for people who are watching their calories, or for people who need to reduce their calorie intake, they are advised to replace sucrose with artificial sweeteners. Now, these sweeteners, they are 600 to 20,000 times sweeter than sucrose, but they are calorie free. So you can take them in limited amounts and you don't have to worry about the calories. But a disclaimer, it is not for everyone. It has to be specifically prescribed by a doctor, and no one should go on taking them or including them in their diet on a regular basis, because they do cause a lot of metabolic disturbances or hormonal disturbances as well. Understood? Next, diseases associated with carbohydrate metabolism. So, where will we study them? In glycobiology, right? So, diseases associated with carbohydrate metabolism, we'll study them in glycobiology. Now, what are these diseases? Diabetes mellitus, right? It is a metabolic syndrome where there is insulin resistance, or there is low or no production of insulin. So, there is impaired glucose metabolism. Glucose doesn't enter inside the cells for utilization, and there is a increase in the blood glucose levels. Okay, next is galactosemia. Galactosemia is galactose is not processed because of the absence of a few enzymes in galactose metabolism. Okay, like galactokinase. Okay, or different other enzymes. Okay, galactose 1 phosphate uridyl transferase. All these enzymes, most of these enzymes are absent. So this uridyl transferase is the most common type of galactosemia. Next is glycogen storage diseases. There are some problems in the storage of glycogen in the liver, so they affect the overall status of the individual, and they are kind of syndromic conditions. Fructose intolerance, where the person is not able to tolerate fructose, that is just after the intake of fructose rich foods like fruits. What happens is they get GI upset, bloating or diarrhea or vomiting and something like that. Lactose intolerance, that is just after the intake of foods containing lactose, that is mostly dairy items, they get a GI upset or bloating or diarrhea or constipation or vomiting and the like. Okay. Now, what are saccharides? Saccharides are aldehyde or ketone derivatives of polyhydric alcohols. Okay, so what is the meaning of this? So there is something called alcohol which has more than one hydrogen atoms. That is, the alcohol molecule has more than one hydrogen molecules, that is on different carbon atoms, and they have an aldehyde or a ketone group attached to the structure. So those are saccharides. Okay, the first is monosaccharides, those saccharides that cannot be hydrolyzed or broken down into simpler sugars. Okay. Hi, good evening, Mudit. Hello. So the first is monosaccharides, which cannot be hydrolyzed or broken down into simpler sugars. That means these are the simplest forms. Trioses means they contain 3 carbon atoms. Tetroses, they contain 4 carbon atoms. Pentoses, they contain 5 carbon atoms. Hexoses contain 6 carbon atoms. Heptoses contain 7 carbon atoms. Understood? Now, what are aldoses? Now we read here that saccharides are aldehyde or ketone derivatives of polyhydric alcohols. So if this saccharide is an aldehyde derivative, it is aldose. That is, the CO, the carbonyl group, is present as a terminal aldehyde. if this polyhydric alcohol is a ketone derivative, that is the carbonyl group is present as a ketone, that is not a terminal aldehyde, rather a ketone, then it is a ketose. Understood? So this polyhydric alcohol, if it is a derivative of an aldehyde, it is an aldose. If it is a derivative of a ketone, then it is a ketose. Classic example of aldose is glucose, and another classic example of ketose is fructose. Okay. Now, if you see BSc second year का class, oh ma'am बता दीजिए. Hi Mudit, this is for the first professional year, the first year in MBBS professional exams. Okay, so but if you want to attend it, you can, because most of the topics you would be reading in your BSc exams also, so you can read whichever topic you find applicable or essential. You can attend the classes. Okay, now trioses, that is the the saccharides containing just 3 carbon atoms. There are just 2 possibilities for these trioses. One is an aldose, that is glyceraldehyde, and one is a dihydroxyacetone, that is ketose. Now, let me know in the comment section, where have we come across both these terms? Glyceraldehyde and dihydroxyacetone, extremely important biomolecules, and we have come across them in one of the cycles, one of the basic cycles of carbohydrate metabolism. Do let me know where and which step. Okay, so there are 2 trioses that are present. One is an aldose, that is glyceraldehyde. What is the meaning of triose? This glyceraldehyde contains 3 carbons. What is the meaning of aldose? This glyceraldehyde is an aldehyde derivative of polyhydric alcohol. Similarly, what is the meaning of triose? This dihydroxyacetone contains 3 carbons. What is the meaning of ketose? That this dihydroxyacetone, it is a ketone derivative of polyhydric alcohol. I hope you guys know that ic or acid, it refers to carboxylic acid. This suffix, it refers to carboxylic acid in organic chemistry. On, it refers to ketone, like dihydroxyacetone. Okay. Dehyde or hyde, okay, it refers to aldehyde. Okay, and all, it refers to alcohol. These suffixes. Okay, so even if you don't remember what glyceraldehyde is, you know that it is aldehyde. It is hidden here, and acetone is hidden here in dihydroxyacetone. Okay, next, what are polyols? So we heard about aldoses, we heard about ketoses. What are polyols? Now, aldehyde or ketones that are present in these saccharides, they can be reduced to alcohols. So already this saccharide is a polyhydric alcohol. Already it contains, now if you see the molecule, the straight chain structure of glyceraldehyde, it is like this. So already it has an alcohol group OH here. Okay. Now, if this aldehyde group is also reduced to alcohol, the CHO also becomes CH2OH here, then this structure will be known as a polyol. Why? Because there will be multiple ols here. Poly means multiple, all means alcohol. So there will be multiple alcohols here, OH, OH, OH groups. So where the aldehyde and the ketone part in this saccharide is reduced to alcohols. Now I hope you remember, I hope you know this. Alcohol is oxidized to aldehyde or ketone, which is then oxidized to carboxylic acid. I hope you know this. And similarly, acid is reduced to aldehyde or ketone, and aldehyde or ketone are reduced to alcohol. So aldehyde or ketones reduced to alcohols present in compounds called polyols. Now polyols can naturally occur in foods also, or they can be artificially synthesized also by reducing the aldehydes or ketones of the saccharides. Okay, now these polyols, they cannot be absorbed after digestion, okay, because we don't have the proper absorbing mechanism. So therefore they will be present in the they will be forming the bulk of the food without them getting absorbed for energy production. So you will be eating, you will be feeling full, but that will not contribute to any calorie production. Okay, so it is used for weight loss. Also, when you want to feel full, that is, you want to increase your bulk in the diet. Okay, so there also it is used. One side effect of this is bloating. Why? Because the polyols will keep lying in the intestine for a long time, and they can be fermented by the bacteria. So bacteria of the small intestine can very nicely act on the polyols. It can break down the polyols, and that can lead to a lot of gas production. Okay, so that can lead to bloating. Okay, but human enzymes cannot, that is, but polyols cannot be absorbed. Okay, one example, do you know of that? Sorbitol. Sorbitol is present in artificial sweeteners. So sorbitol, you take sorbitol, and that sorbitol is not going to get absorbed, but it is going to be sweet in nature, but it is not going to get absorbed, so it is not going to contribute to calorie production. Understood? Next. So we are over with monosaccharides. Monosaccharides can be divided into trioses, tetroses, and the like, depending upon the number of carbon atoms. Monosaccharides can be divided into aldoses, ketoses, or polyols. Next, we come to disaccharides. Disaccharides are the condensation products of two monosaccharide units. Di means two. So two monosaccharides will combine to form one disaccharide. Lactose is formed by the condensation of glucose and galactose. So glucose plus galactose will produce lactose. Now maltose is produced by the condensation of two glucose units, glucose plus glucose. And sucrose is produced by the condensation of, anyone in the comment section? Glucose plus one very important biomolecule, one very important saccharide, which is fructose. Understood? So lactose is glucose plus galactose. Maltose is glucose plus glucose. Sucrose is glucose plus fructose. Isomaltose is an isomer of maltose. Okay, understood? So monosaccharides we have read about, and we have read about disaccharides. Next we come to oligosaccharides. Okay, now so oligosaccharides, now disaccharides contain two monosaccharide units. Oligosaccharides contain 3 to 10 monosaccharide units that are condensed together. Okay, most oligosaccharides are not digested by human enzymes, and they encode important biological information. So you'll read about a few important oligosaccharides later on. They are short chain saccharides that perform different functions other than the nutritional function, because most of them are not digested by human enzymes. Okay. Now, what are polysaccharides? Polysaccharides are those saccharides that are the condensation products of more than 10 monosaccharide units. So it is 2, then 3 to 10, and then more than 10. So important polysaccharides: starches, dextrins, cellulose, inulin, glycogen, chitin. So these are the important polysaccharides. Okay. There are starches, that is starch, dextrins. Okay. What are these starches? They provide energy, or they are absorbed for nutritional purposes. And there are non-starch polysaccharides that act as dietary fiber. Why? Because they are not broken down by human enzymes and they form the bulk of the diet. They form the bulk of the food that passes through the intestine, and they're only to add fiber or bulk to the food to make you feel fuller and to avoid constipation. Understood? So, we'll start with the most important biomolecule, that is glucose. Now, the structure of glucose can be represented in three different ways. Okay, first is the straight chain formula. As we know, it is an aldohexose, that is, it is an aldehyde derivative of a polyhydric alcohol, and it contains 6 carbon atoms. So, glucose is an aldohexose, and the straight chain formula is depicted like this. So 6 carbon atoms you make like this. So this one is CH2OH. Okay. And here this one is the terminal carbon is CHO. Got it? This is first carbon, second, third, fourth, fifth, and sixth. Okay. So now on the third carbon, the location of H and OH is different from the rest of the carbons. So it is like this. On the third carbon, it will be like this. Then again like this. So this is the straight chain representation, okay, of glucose. Next is the cyclical structure. Now, cyclical structure can be either the Haworth projection or the chair projection. So there are total 3 ways in which the structure of glucose can be depicted. Either a straight chain formula or cyclical structures, okay. Now, if you read or if you make the cyclical structure, then you can account for most of the properties of glucose. Okay. So always learn how to draw the cyclic projections. Okay. One is the Haworth projection. Here you draw O like this because it is a 6 carbon molecule. So you draw it like this. Now this is a hexagon, but on one of the vertices you have O. So where do you keep? So you will have only 5 carbon atoms. That is, this is 1, this is 2, this is 3, this is 4, and this is 5. So where will the 6th carbon be? The 6th carbon is like this here, CH2OH. Now you're looking at this ring from above and from the side. So these bonds will be darkened here. These bonds are darkened. Understood? And you don't mention the H atom in the Haworth projection, okay? Because it will lead to a lot of crowding. So you only mention OH. And it is assumed that this is H, this is H. Now third pay, it is opposite. So OH will be above. Now this is again OH, this is again OH. Understood? So this is how the Haworth, this is the straight chain. This is the Haworth projection. And the last will be the chair projection. How is the chair projection? Now you draw it like this, like a bow. Okay, you draw it like a bow. Okay, so this has to be in this way, sorry. So this is drawn like a bow. Got it? So this is the chair projection. So this is like a chair. Understood? Here you draw this also. So this will be O. Okay, this here is O. So this same cyclical structure, you have kind of twisted it to resemble a chair. So this is a chair, right? You can assume someone sitting like this over here. So this is a chair. Okay, so this is the chair projection. And here you will have CH2OH. You draw H also here. So this is 1, 2, 3, 4, 5, and 6. Okay, so HOH also you have to draw here. Now I hope you understood all the three different ways in which you draw the projections. Okay, so H and OH, this is the first one. This is the second one. Again, you draw H and OH like this. The third one, which is different, the OH will go on the other side. Now here again, H and OH. Here again, H and OH. So this is how it is. This is the chair projection. I hope you can draw all of this. Just remember, you have to draw a hexagon. On one of the vertices, you have to put oxygen so that the last carbon atom is hanging above, okay, from the fifth carbon atom. Also, you go about in a clockwise manner just after the oxygen to number the carbon atoms. The third carbon atom is behaving differently. So OH will be on the opposite side in all the three projections. This is very easy, very, very easy. Okay, if you have any problems, you can let me know. I'll tell you how to draw again. Okay, now the types of isomerism that glucose exhibits. Now, glucose has 4 asymmetric carbon atoms and therefore it can form 16 different types of isomers, right? 4 asymmetric carbon atoms. How? This is one asymmetric carbon atom. This is one asymmetric carbon atom. Any of these can be an asymmetric carbon atom, and this is one asymmetric carbon atom. Asymmetric means different from the other. So out of the 6 carbon atoms of glucose, it has 4 different or asymmetric types of carbon atoms that are different from each other. Okay, right? Therefore, it can form almost 16 isomers. The first type of isomerism is capital D and capital L isomerism. Now, this is accounting for the spatial relationship to the parent compound, which is the triose, the aldotriose. Why? Because glucose is an aldehyde or glucose is an aldose. Therefore, the parent compound of this aldohexose will be the simplest aldose, that is aldotriose, right? So, aldotriose is what? Glyceraldehyde. Aldoketose is what? Dihydroxyacetone. So, the parent glyceraldehyde, here if we see CHO, C and CH2OH. Now, if the carbon atom adjacent to the terminal alcohol, if the carbon atom adjacent to the terminal alcohol has OH on the right side, then it is D-glyceraldehyde. And if it has OH on the left side, then it is L-glyceraldehyde. Similarly, for glucose, if the carbon atom adjacent to the terminal alcohol, that is this one, has OH on the right side, this one, if this has OH on the right side, then this is D glucose. And if suppose this OH had been on the left side, if suppose this chain would have been like this, now if suppose here everything had been on the left side, apart from the third one, because the third one behaves differently, right? So if suppose this OH had been on the carbon adjacent to the terminal alcohol, if this OH had been on the left side, then it would be L-glucose. Understood? Now, plus and minus isomerism. What is plus and minus isomerism? Here it is optical isomerism. That means stereoisomerism. That means if a beam of plain polarized light is passing through a solution of either plus or minus isomer, then if the solution rotates the light to the right side, it is dextrorotatory, or if it rotates it to the left side, it is levorotatory. So it can either be D plus or D minus, or it can be L plus or L minus. Earlier, this plus minus was represented by small d and small l, which led to a lot of confusion between D and L, capital D and capital. So now they've changed the nomenclature to plus and minus. Understood? Next, pyranose and furanose ring structures. Now, pyran is a 6-membered ring. It is like this. Okay, pyran is a 6-membered ring like this, and furan is a 5-membered ring. Double bond, double bond, double bond, double bond. Got it? So this is a furanose, that is a 5-membered ring. This is pyranose, that is a 6-membered ring. You know that glucose is pyranose. That is almost 99% of glucose is pyranose. Next, alpha and beta anomers. What are anomers? In the ring structure of aldose, that is hemiacetal form. Acetal means reaction between aldehyde and alcohol. So here, this aldehyde and this alcohol reacts, right, in the ring structure. So the ring structure is a hemiacetal form. Here what happens is, here whenever there is the presence of OH or H is present above or below the structure of the ring, then you have another type of isomerism, and you have alpha and beta anomerism here, that is also in ring type of structure. Okay, so ring ketose is hemiketal and ring aldose is hemiacetal. What is hemiacetal? Reaction between aldehyde and alcohol. Reaction between ketone and alcohol is hemiketal. Okay, crystalline glucose we know is D-glucopyranose. In solution, the cyclic structure is retained, but isomerism occurs about the first carbon. Okay, the first carbon is this one. Okay, here 38% is alpha glucopyranose and 62% is beta glucopyranose. So maximum glucose is beta glucopyranose. Okay, now what are epimers? Epimers are isomers differing as a result of configuration variations of the OH and H on carbon atoms 2, 3 and 4 of glucose. Okay, so this is glucose. So this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4. This is the first carbon OH, H. This is the second carbon OH, H. This is the third carbon OH, H, OH, H, OH, H and CH2OH. Now this is glucose. Okay. Now in galactose, this is 1, 2, 3, 4. This fourth here, what will happen is this will be twisted. So what will happen here in galactose is it will be upside down. Galactose will be OH and H like this. The rest will be same. And what will be mannose? Mannose will be like this. 1, 2. This one will be OH and H. Got it? Now this has turned upside down. So these are epimers, that is differing in the configurational variations of H and OH on carbon atoms 2, 3 and 4 of glucose. Aldose ketose isomerism we know. Fructose and glucose both contain 6 carbon atoms. Both are hexoses. But Fructose is a ketose. Glucose is an aldose. Okay, fructose is a furanose ring. Glucose is a pyranose ring. Okay, now aldoses are reducing sugars. Okay, that means they reduce. Okay, so what happens is whenever glucose, which is an aldose, whenever you pee on a pee stick, on a urine dipstick test, whenever you pee on that, what happens is it contains a solution of copper sulfate, or it contains a copper solution that is reduced by this aldose, and there is color change that is occurring depending upon the concentration of glucose. So urine strip test is used for that. Now, what are the properties of monosaccharides? Okay, monosaccharides, they are intermediates of glycolysis. We know that there are a lot of intermediates. Trioses we have already learned about. Glucose, fructose, all these are intermediates of glycolysis. We'll read about them later on. They are intermediates of HMP shunt. Now, trioses, tetroses, hexoses, they are intermediates of the HMP shunt also. Pentoses, they form part of nucleic acids. Monosaccharides are constituents of glycoproteins as well, okay, especially arabinose and xylose. Monosaccharides like xylulose are excreted in the urine in essential pentose urea. So these are very important properties of monosaccharides, okay. Now, what are glycosides? Glycosides are condensation products of the hydroxyl group of anomeric carbon of monosaccharide with another monosaccharide or a non-monosaccharide. Okay, so what happens is glycoside means any bond that is formed with the anomeric carbon. What was the anomeric carbon? This, the first carbon. This is the anomeric carbon. This one. Either the OH is on top or OH is below. Okay, so alpha is when OH is on top, beta is when OH is below. So here what happens is the bond, the condensation between the OH group on this carbon with another monosaccharide is called glycone, where the monosaccharide is called glycone, or with a non-monosaccharide, that is, a non-monosaccharide can be an amine, that is NH2 group. And there it is called an aglycone. So the hydroxyl group of glucose is forming a bond with the hydrogen group of another saccharide, or it is going to form a bond with NH2. This is aglycone and this is glycone. Okay. If the second group is also a hydroxyl group, if this is also a hydroxyl group, then an acetyl ring link is formed. Okay. Like in glucoside or galactoside. Glucoside means two glucose molecules are forming a bond. So this is a glucose molecule. Okay, this is OH, this is H, this is another glucose molecule. This is H, this is OH. So if both OH are reacting together, then what will happen is a CHO group will be formed, that is an acetyl link. So this is a glucoside. Or if the second group is an amine, suppose if this is an NH2 group, then an N-glycosidic bond is formed. N, that is nitrogen is also present here. N-glycosidic bond is present in adenine and ribose in nucleotides. That is in nucleotides, you know, nitrogenous bases, N, they are present. Understood? So anomers, very, very important. So what are different aglycones? That is non-monosaccharide entities that form the second part. They can be methanol, glycerol, sterol, phenol, adenine, or whatever. Understood? So very important, glycosides. So different monosaccharides interact together to form glycosidic bonds. Glycosidic bonds to form disaccharides or polysaccharides and whatever. Okay, so the bond between them is called glycosidic bond. Now, important glycosides, that is important condensation products using glycosidic bond, are cardiac glycosides like digitalis or uabane. They contain steroid aglycone. That is the second reacting element is a steroid. Uabane inhibits the NAK ATPase enzyme. Antibiotics like streptomycin are also glycosides. Okay. Now, There are certain deoxy sugars where an oxygen atom is removed. Okay, so the hydroxyl group is replaced by a hydrogen group, right? So you know there are a lot of hydroxyl groups here. So one of the hydroxyl group, if it suppose this OH is replaced by an H, okay, then it is known as a deoxy sugar. Very easy. O is removed, oxygen is removed, like deoxyribose in DNA, L-fucose in glycoproteins, 2-FDG in PET scan, right? 2-FDG is what? It is taken up by very highly by tissues that are very active in metabolism, that have a very high degree of metabolism, because it is a form of glucose, and wherever they are taken they cannot be metabolized further. Okay, fluorodeoxyglucose, 2-fluorodeoxyglucose, it is not metabolized further, and then it can be picked up on PET scan. So metabolically active tumors or metastasis can be visualized using injection of 2-FDG and then exposing them to PET scan. So it is a very important application of a deoxy sugar, that is 2-fluorodeoxyglucose. Okay. Now deoxyglucose is fluorinated at the second position with fluorine. Now it cannot be acted upon by enzymes. It cannot be taken up for energy production. Therefore, it is accumulated. Understood? Now comes amino sugars. As the name suggests, they contain an amino group. And now amino group they will be components of glycoproteins. Amino is synonymous to proteins. They're components of gangliosides. What are gangliosides? Now they also contain glycosidic bonds and glycosaminoglycans like D-galactosamine. It is present in cartilage, chondroitin. Antibiotics, that is erythromycin, contains amino sugar. So one antibiotic, that is streptomycin, it contained glycosides. Another antibiotic, that is erythromycin, contains amino sugar. Okay, so this is very important. In your next year in pharmacology, you're going to read about them. So your basics should be very clear. Biochemistry should be clear for you to master pharmacology as well. Okay, understood? Next, now we are over with monosaccharides, the important disaccharides. So two monosaccharides linked by a glycosidic bond, as I already explained, is what is forming a disaccharide. Okay, now maltose, lactose and sucrose. Quickly tell me, maltose is a disaccharide of glucose and glucose. Lactose is a disaccharide of glucose and galactose. Sucrose is a disaccharide of glucose and fructose. Okay, so sucrose on hydrolysis is going to give glucose and fructose. Why is that called invert sugar? Because fructose is very strongly levorotatory. Okay, it rotates the plane polarized light to the right, whereas the initial sucrose to begin with was dextrorotatory. So it changes the property of sucrose to levorotatory, therefore it inverts it from dextro to levo. Therefore fructose here is known as invert sugar. Okay, understood? It is also very important. You're going to get a short note on invert sugar, maybe a definition or something. There you're going to explain about optical isomerism. You're going to explain about the structure of fructose and glucose and the structure of sucrose, and how sucrose on hydrolysis is going to form glucose and fructose, where fructose will change the optical isomerism of sucrose from dextrorotatory to levorotatory, because it is very strongly levorotatory. Therefore, it is called invert sugar. Okay. So this was all about the important disaccharides. Now we come to important polysaccharides. Okay, what is the function of polysaccharides? They provide structural and mechanical protection. They are there for energy storage, structural and mechanical protection, like glycosaminoglycans, okay, proteoglycans, and the like. Energy storage in the form of glycogen, starch, and they bind water to resist dehydration, like inulin. Okay, understood? And starches and gums and all that. Okay, so we are going to read about different polysaccharides. Now starch, the most important. Starch is a homopolymer of glucose. That means more than 10 glucose subunits Homo means only same type of monosaccharide is going to come. Okay. So more than 10 glucose molecules are going to join together by glycosidic bonds. Okay. To form a chained structure that is starch. Now, there will be glucose, glucose. This is a straight chain. Understood? And what is this bond between these glucose molecules? What is this bond? This is alpha 1,4 linkage. That means the first carbon of this is attached to the fourth carbon of this. It is very easy. If you draw the cyclical structure of glucose, okay, this is the first carbon. This is attached to the fourth carbon here of the next glucose. 1, 2, 3, and 4. This is the fourth carbon. So they form the glycosidic linkage here. So this is alpha 1,4 linkage. Okay. Why alpha? Because this is the alpha anomer. Why alpha anomer? Because OH is present on the top. Now, the branches here occur at alpha 1,6 linkage. That means 1, 2, 3, 4, 5, and 6th. So the 6th carbon here, if you branch, the 6th carbon, that is CH2OH, of another glucose here, this. So this is going to form bond with the first carbon of this glucose. So alpha 1,6 here is what? This is the branch, and this as it is going to continue. The chain is going to continue and it will be interrupted by frequent branches. Okay. So what happens is amylose is the non-branching helical form like this. It is the non-branching form of starch. It has alpha 1,4 linkages. And amylopectin, which forms the major part of starch, it is the branched form also. That means it has alpha 1,4 straight chain linkages and alpha 1,6 branched chain linkages also. So starch consists of 13 to 20% amylose. Amylose is straight or non-branching helical structure having alpha 1,4 glycosidic linkages between different glucose molecules. And starch contains 80 to 87% amylopectin. Amylopectin is the branched form where the straight chains 24 to 30 glucose residues have straight linkages, alpha 1,4 linkages in the chain, and there are interrupting side chains or interrupting branch points, and the branch points are formed at alpha 1,6 bonds. Understood? Now, the extent of hydrolysis of starch by the enzyme amylase, that depends on the structure, the degree of crystallization or hydration of starch, that means the amount of cooking that the starch has been subjected to, and whether it is enclosed in intact cell walls. Why? Because cell walls are resistant to human enzymes. So if the starch is intact inside a cell wall, then it is not going to get digested or it is not going to get hydrolyzed by amylase faster. Therefore, this is what brings us to the glycemic index. Okay. So if amylase is able to break down starch very quickly and it raises the blood concentration of glucose very quickly compared to an equivalent amount of glucose or white bread or boiled rice, which is the reference, then the glycemic index of that starch is said to be very high. So suppose you are having a cooked meal of potato. Okay. Now that cooked potato, it is very easy to digest. Okay. Now amylase will act on it and it is going to quickly break it down and the blood sugar will rise very quickly. It will shoot up. Okay. So if the blood sugar rises very quickly compared to when you are taking a glucose meal, then the glycemic index of that meal is going to be very high. Whereas if the glycemic index of a starch is low, that means that starch is digested very slowly and the glucose that is released in the blood is at a very small rate. The glucose concentration does not peak suddenly, it is almost plateauing. Okay. So the glycemic index ranges from 1, that is 100% or readily hydrolyzed, to 0 for those that are not hydrolyzed. So 1 is very high glycemic index and 0 is low glycemic index. So glycemic index also forms part of your short notes. So please read about glycemic index as well. Next, glycogen. So we have read about starch. Glycogen, starch was the energy storage polysaccharide in plants. Glycogen is the energy storage polysaccharide in animals. Therefore, it is known as animal starch. Glycogen is present in the liver or in the muscles. Okay. Now, glycogen is highly branched. It is more highly branched than amylopectin. So, starch contained unbranched amylose and branched amylopectin. Glycogen is even more branched than amylopectin. It has chains of 12 to 25 alpha D glucopyranose residues. And those pyranose residues, as in starch, they're going to form alpha 1,4 glycosidic bonds, and branching will occur at alpha 1,6 linkages, similar to amylopectin. But what is the difference? In amylopectin, the branching was rare. Here, the branching is more. So alpha 1,6 linkages are more frequent. Understood? So this is the difference between glycogen and starch. Next, inulin. Inulin is extremely different from insulin. Insulin is a protein. Inulin is a saccharide. Inulin is a fructose derivative. It is derived from fructose. Now, therefore, it is called fructosan. Okay, now the tubers and the roots of plants like dahlia, dandelion, and artichokes, they contain inulin. Inulin is not hydrolyzed or digested by the human intestinal enzymes. Therefore, it does not have any nutritional value. It is not going to provide any calories. It is only used to form the bulk of the food, that is dietary fiber. One important application of inulin is in the assessment of glomerular filtration rate in the renal function test. The rate at which it is excreted, that is measured in the glomerular filtration test. So whenever we are going to read about renal function tests later on, we will touch upon the usage of or the application of inulin. Next comes cellulose. Cellulose is the chief constituent of plant cell walls. Do not forget to include plant cell walls. Okay. Now, it has beta D glucopyranose units. Okay. That means the anomer at the first carbon is beta form. That means OH is below. Understood? So this is beta carbon. Understood? Now, of course, if this is the first carbon is the beta carbon, then 1,4 linkage is going to be beta 1,4 linkages. But humans don't have, or mammals don't have enzymes to hydrolyze these beta linkages. They can only hydrolyze alpha linkages like in glycogen or in starch. Therefore, this cellulose is not digested by mammals. Okay, cellulose, which is a constituent of cell wall of plants, is not digested by mammals because they cannot hydrolyze beta 1,4 linkages. But cellulose can be metabolized by bacteria, and bacteria that is present in the human gut, it metabolizes or it digests cellulose to form short chain fatty acids. And these fatty acids, they have a role to play in our gut health, in the immune system of the gut. And with the rising trend of probiotics and prebiotics, we can change these the bacterial population inside our gut to form different types of short chain fatty acids using cellulose, and that is going to contribute to the immune system or immune health of the associated with the gut. Okay. Now chitin forms a part of fungal cell wall and exoskeleton of crustaceans and insects. Chitin has N-acetyl D-glucosamine units linked by beta 1,4 linkages. Again, beta 1,4 linkages here also, and N-acetyl D-glucosamine units. Okay, understood? Now it has amine. Amine means That means it has NH2 group. Okay, so it does not just have an OH or an H group on the second carbon, it has an amine group on the second carbon. So it is an A glycon. Remember, this is what we read about the glycosidic linkages. Okay, now pectin. Pectin is present in fruits like guavas, and it is a polymer of galacturonic acid. That is an acid linked by alpha 1,4 linkages with galactose or arabinose branches, and it has partial methylations. Now we come to glycose aminoglycans. Glycose aminoglycans can also be called mucopolysaccharides. You will have a separate class for them, which is an extremely important unit in biochemistry and very interesting, very easy to know. So these glycose aminoglycans, okay, so glycan is also saccharide, glyco is also saccharide, and amino is something related to protein. Okay, so they have something related to, they are something related to saccharides and proteins. They are units of amino sugars and uronic acids. Understood? And if they are attached to proteins, then what will they be called? They will be called proteoglycans. Of course, glycan is what? Sugar. And proteo is what? Protein. A very important and a very trending glycosaminoglycan is what? Hyaluronic acid, present in a lot of, in almost every cream and serum you will have hyaluronic acid serum or hyaluronic acid based cosmetics. Then chondroitin sulfate present in cartilage. Then heparin, which is an important anticoagulant present in humans. So they are all proteoglycans and glycosaminoglycans. Next is glycoproteins or mucoproteins. So those were proteoglycans. Proteoglycans means they are actually, the surname is glycan. So you can have different names. So you, your brother, your sister will have different names, but you will have same surnames. You belong to the same family. So if you have the same surname, you will belong to the same family. Your names can be different. Similarly, the surname here is proteoglycan. So which family do they belong to? Glycan family, that is saccharide. So they are essentially saccharides which have a protein group attached. But what is the surname here? It is protein. So which family do they belong to? The family of proteins. So these are essentially proteins with a branched or an unbranched oligosaccharide chain attached, like fructose. You know fructose, we read about fructose in the deoxy sugars. Deoxygalactose is what is called fructose. Deoxyribose is present in DNA. Understood? Next are sialic acids. What are sialic acids? They are N-acetyl derivatives of neuraminic acids. What are neuraminic acids? 9 carbon sugar. So neuraminic acids are 9 carbon sugar derived from mannose-amine. What is mannose-amine? Amine and mannose. That means they will have something called NH2 group and they will be a derivative of mannose. So mannose amine, like mannose is an epimer of glucose. Similarly, mannose amine will be an epimer of glucose amine. Mannose is an epimer of glucose at which carbon? Do let me know in the comment section. And galactose is an epimer of glucose at which carbon? Do let me know in the comment section. We have already covered it. Okay, these sialic acids are constituents of glycoproteins and gangliosides. We already know that. Next is glycated proteins. So we know about proteoglycans. Proteoglycans kya surname hai? Glycan. That means they are saccharides. Their family name is glycan. Glycoproteins. Surname kya hai? Protein. So surname is, what is their family? What is their family? Protein. Glycated protein. What is the surname? Protein. So what is their family? Protein. So whenever there is an addition of a saccharide to a protein through a non-enzymatic mechanism, then it is called glycation. And if it is through an enzymatic mechanism, it is what is glycoprotein. Okay, a classic example is by glucose. And that is this HbA1c present on the RBC membrane. This HbA1c is glycated by a glucose molecule, and this RBC survives for around 120 days. Therefore, more the glucose in the blood, more will be the degree of glycation, and higher will be the HbA1c that is measured in the blood test. Therefore, HbA1c is used to monitor the level of glucose control in diabetics. Also, it is used to diagnose diabetes by the American Diabetic Association. There are different guidelines for that. Okay, understood? So this is the application of HbA1c. Next are glycocalyx. Okay, so what are glycocalyx? Glycocalyx is an important component of the outer surface of plasma membrane. You know that the plasma membrane contains phospholipid, a bilayer of phospholipid, right? And the outer surface, a phospholipid bilayer is there. The outer surface has a few saccharide molecules which are known as glycocalyx. What are glycophorins? They are integral glycoproteins in RBC. Integral glycoproteins means they are present inside the RBC. Glycoprotein means what are they? They are proteins. Surname is protein. So there should be no confusion between proteoglycans, glycated proteins, and glycoproteins. Okay? If you have a confusion, go back and revise this class. So, glycophorin is an integral glycoprotein in RBC that forms a channel. Okay. Now, ABO blood grouping also depends upon a few saccharides. Okay. What happens is, now ABO blood groups is distinguished by the presence of a particular enzyme in the genome of the person. Okay, the enzyme that is encoded by the genome of the person. Suppose someone has A group of blood. Okay, the blood group is A. That person, the genome will code for A transferase enzyme. Okay, and that A transferase enzyme is going to then attach an oligosaccharide, that is N-acetyl galactosamine. This galactosamine, you know, it is an epimer of glucosamine, just like mannose-amine was an epimer of glucosamine. So A transferase is going to attach N-acetyl galactosamine to the membrane of RBC. And whenever N-acetyl galactosamine is present on RBC membrane, then that RBC is A blood group. If someone has a B transferase enzyme, that enzyme is going to attach galactose on the RBC membrane. And whenever galactose is attached to the RBC membrane, the blood group is B blood group. This is that easy. So these are what oligosaccharides, N-acetyl galactosamine, galactose. All these are oligosaccharides that are attached on the RBC membrane. For O, there will be an inactive ABO glycosyl transferase enzyme, and therefore no oligosaccharide will be attached on the RBC. So isn't it very intriguing and fun? You know how, now you saw how varied and how diverse is this chapter. Therefore, that is why we say that saccharides are the most abundant biomolecules. You have touched almost the whole of biochemistry while reading about just the introduction of carbohydrates. Therefore, carbohydrates or saccharides are the most abundant biomolecules because they are present in almost every sphere of biochemistry. Okay, so we have covered glycosaminoglycans, we have covered proteoglycans, glycoproteins, glycated proteins, blood grouping, cell membrane, cell wall, nutrition, digestion. We have completed neurotransmitters. We have touched with so many different areas just by reading the introduction of saccharides. So very important take home for you from this is that you have to read the applications of saccharides. You should know the different types of isomerism, especially anomers, epimers, D and L forms, plus and minus forms. All these things you should know. What are the differences? Then you should know how to draw the three structures of glucose. You should know the different types of glycosidic linkages in different types of polysaccharides. You should know what a glycosidic linkage is. You should know what is the difference between a glycone and an aglycone in glycosidic bond. So all these important, these are the most of the take home factors from this. Okay. Now after this chapter, we are going to start with the metabolism of biomolecules. We're going to start with an easy topic that you have already read in class 12th, that is glycolysis and Krebs cycle. And I have already made a video of that using mnemonics and a story based explanation. So please, if you haven't watched that video, go back to the channel and watch the video of Krebs cycle made easy and glycolysis made easy. And we are going to read these two topics tomorrow in the at the same time, that is at 6 p.m. tomorrow on the same channel. Okay, so stay tuned to that. Please, please, please keep on revising. Just watch these videos on 2x. Okay, whenever you're revising, watch these videos on 2x. Just jot down your notes and please get into the habit of making the diagrams as you read. Okay, you don't have to wait for just one day before the exam. Just make diagrams as you read. Okay. All right, guys, then thank you so much. It was an interesting session. A lot of diagrams, a lot of writing down, and loaded with chemistry. Okay. So next we are going to meet tomorrow with glycolysis and Krebs cycle. Okay, guys. Bye-bye. Take care.